So today we're going to talk about the general structure of ovaries and a couple of other things. So first of all, what are ovaries? So the ovaries are the organs, the first organs that house the um, ovums, and they're almond in shape. And thing is, each ovary is covered in germinal epithelium, also known as simple cuboidal epithelium. And like we know, and the same goes even for the testes. The ovary is also covered in a thick, dense connective tissue layer known as the tunica albuginea. Now, the thing is, the ovary is divided into two main parts, like you can see in the picture here. Okay, it's not very clear, but you can see it here kind of. There's a okay, yeah, so there's a medulla and there's a cortex. So the difference between the medulla and the cortex is that the medulla is loose connective tissue. It consists of like blood vessels and it has like loose connective tissue. So it's like the main part, the medulla, whereas the cortex is just the surrounding stroma. Stroma is basically like supporting cells that support this medulla. And like for example, like ovum has supporting cells around it, known as the ovarian follicles. So this is so you will see the ovarian follicles in the cortex of the ovary. Now let's talk about the early development of the ovary. In simple words, the easiest way that I've written it down here is in the form of a flowchart. So the ovary itself, it comes from the primordial germ cell that's around the time of fertilization. Like we know how fertilization takes place when an ovum and a sperm uh, meet and they form a egg. So what happens is we have the gonadal primordia. So the gonadal primordia is essentially cells that form the ovary. They undergo several mitotic divisions, like several incomplete mitotic divisions. And because of this, what happens is that they're interconnected. Like I mentioned, they're incomplete mitotic divisions because of which they are all interconnected, and now this interconnected oogonia gets arrested first in the prophase 1 of meiosis 1. And what happens is most of them, many of them, I would say, undergo apoptosis, and it results in the death. And some of them, they complete their genetic recombination that takes place in the meiosis 1, and they actually end up forming the primary oocytes. Now these primary oocytes are essentially cells. They are actually structures that are surrounded by the supporting cells known as the follicular cells that actually end up forming the ovarian follicles, right? And another uh, like just a general fact is that the ovarian follicles at birth there's a very high amount of like ovarian follicles, but not at birth, sorry, when yeah, so the ovarian reserves, this is what I was trying to say. Sorry, yeah, excuse me. So the ovarian reserve actually reduces with time. So at birth, it's very high, and at puberty, it reduces a bit more. And around lifetime, we're just left with 450 to 500 eggs, which is okay because every month, there's only a release of one egg. So the 400 to 450 to like 500 eggs are enough to sustain for a lifetime. So that's one thing. The next thing we're going to talk about is we're going to talk about the follicles. So the ovarian follicles, as we know by now, are the supporting cells that we've come across. They're seen in the cortex of the ovary. So the ovarian follicles actually cells that surround the oocyte, the primary oocyte. And the thing is, these that's all that is their main function, and we end up seeing like different like ovarian follicles, like we see the primary follicle, the secondary follicle, the tertiary follicle. We'll talk about it in the as we go ahead. A characteristic feature of oocyte is just the fact that it is spherical and it has a 25 micrometers diameter with a very large nucleus. Another thing we need to talk about when we're discussing the ovarian follicles is simply we need to mention the blood follicle barrier. So we by now we know about different kinds of like blood like barriers. For example, we have a blood air barrier in our lungs, we have a blood brain barrier in our brain, we have a blood testis barrier, I think, in the male genital system and female genital system. We have the blood follicle barrier. So basically, the blood follicle barrier ensures that um, it primarily ensures it's just a barrier, like we talked about. It separates the follicle actually separates. Uh, so what? It actually really is that the basal lamina of the primary oocyte separates the follicle from the vascularized stroma. Right? So that's about it. And then we're going to talk about the follicular growth and development because this is extremely important. And this is a very commonly asked question. This is a very general question that can be asked. So first of all, the follicular growth is stimulated by which hormone? The follicle stimulating hormone, which is released from the anterior pituitary. We're aware of that. And another thing is, like we mentioned, we discussed a few characteristics of the oocytes. So initially, oocyte is around 25 micrometers, but as it grows, right before its release, it increases to around 120 micrometers in size with like a bigger nucleus because it's increasing in size, so its nucleus size is going to increase. The organelles are going to increase, and there's a lot of other things that are going to just increase because of the increase in the size. So now what happens is we're going to talk about the different kinds of follicles that we see surrounding this oocyte. So a primary follicle is a follicle that surrounds the primary oocyte. They can be either unilaminar or multilaminar. So basically unilaminar, so it's like one layer. So like there's an oocyte. Just imagine there's like a circle, there's like an oocyte. It's surrounded by one layer of like supporting cells known as the follicular cells. So there's a laminar uh, primary follicle that is essentially just simple cuboidal epithelium, and the multilaminar um, primary follicle is basically when there are multiple layers of like these follicular cells surrounding the oocyte, and so the simple cuboidal epithelium now changes to stratified follicular epithelium. So that's what we're gonna that's about it. And other than that, we also see like how I talked about. So we have the oocyte, and then we have the first layer of the follicular cells. So between these two, we start seeing the accumulation of extracellular matrix. Because of this, as the extracellular matrix accumulates, we actually end up seeing. Oh wait, I don't know if you can hear me. Wait, I think there was an issue. So yeah. So what was I talking about? I was talking about that between the oocyte and the first granular layer, we see the accumulation of the extracellular matrix, which ends up forming the zona pellucida. Now, zona pellucida is just a layer, another layer that surrounds the oocytes, and it actually has glycoproteins that help in the this is like it has glycoproteins that help uh, when the sperm is trying to penetrate the ovum and mm, just right before fertilization. That's what zona pellucida is. And the thing is, another thing that we notice is that the rest of the cells, so the first layer forms the zona pellucida, and the following layers that are outside of it, what actually happens is that they become vascularized and they form two kinds of cells. So the inner one, so we have the oocyte outside outside of it we have after so we have the oocytes outside of it we have the zona pellucida. Outside of it, we have the theca interna. Outside of it, we have the theca externa. So the follicular cells, the multilaminar follicular cells, they actually became theca interna and theca externa. And like them, they have their own functions. Where theca interna basically, it helps in the secretion of like estrogen, and that's its main function. It's very well vascularized with endocrine because of the secretion of uh, estrogen, whereas theca externa, it has more of like a covering sort of function where it has fibroblasts and smooth muscle cells. And yeah, now further development is like we talked about unilaminar primary follicle, multilaminar secondary follicle. Now these follicles, they move deeper into the cortex, and they actually um, yeah. So another thing we need to talk about is now these follicular layers, as they start becoming secondary follicles, we call them the granulosa layers. So the granulosa like cells, the granulosa cells actually start secreting like follicular liquid, which is is a characteristic feature of the secondary follicle known as also antral follicles, because in the secondary follicles we see the follicular liquid, whereas in the primary follicular follicles we do not. So that's really, really important. And the last but not the least is the Graafian follicle. It's just the thickest one of all. And that's also known as a tertiary follicle. Yes. Now we also need to talk about another very basic concept. It's a very tiny concept. It's something you just need to know at the back of your head. It's known as follicular atresia. Which is basically a degenerative process where the follicular cells, yeah, so the surrounding follicular cells, they actually undergo apoptosis. They actually die by undergoing the process known as apoptosis, right? And in the end, what actually happens is that there is only one dominant follicle, and the rest of the follicles they degenerate. But as they degenerate, we know that they end up producing estrogen because a part of it is theca interna, which is endocrine in function, which produces estrogen. So they produce high amounts of estrogen, and that is what is follicular atresia. So in a nutshell, it's basically that there's one in the event of follicular atresia, which happens is basically there's one dominant follicle and the rest of them degenerate. That's it. Now we also need to talk about ovulation. So ovulation. So what is ovulation? So ovulation is nothing but a hormone regulated process that happens midway through our cycle. So what happens is just before ovulation, what happens is the mature so we talked about how there's follicular atresia and there's one mature dominant follicle in the end. So it develops this whitish spot. It's known as the stigma where the blood flow is blocked so that the oocyte can be released. Yeah, and just before ovulation, the first mitotic division of the oocyte is complete, and the second mitotic division completes only if it meets the sperm, and it ends up it results in the formation of a zygote, right? So I've written it down in a very concise manner. It's basically that the primary oocyte is arrested at the prophase one, yeah, and and once it completes its meiosis one, it forms a first polar body which degenerates and it forms a secondary oocyte. Now this secondary oocyte also gets arrested at metaphase, and just before its release, it forms ovum. Sorry, yeah, the secondary oocyte. Sorry, I'm sorry. Yeah, the secondary oocyte actually it completes its meiosis. So the metaphase two, I should have mentioned it here. Yeah, so secondary oocyte is arrested at metaphase two, and it only completes its meiosis two upon fertilization, which results in the formation of ovum as well as the second polar body, and the polar bodies degenerate, right? So that's the most important part. This negative feedback, it's not important here. It's more of an endocrine thing. So the last thing we're going to talk about has to be the corpus luteum. So we talked about how the stroma cells, the follicular cells, um, once they become multilaminar, we call them the granulosa cells. And then we have layers in it. We have the theca interna and the theca externa. So the corpus luteum naturally has two kinds of cells, the granulosa cells and the theca interna. Theca externa just degenerates, and because of which, as we know, the theca interna secretes estrogen. So this is also an endocrine gland that is present in the ovarian cortex, and it is very well vascularized, I would say. So we have two kinds of cells. They are now these cells. Now the granulosa cells are called granulosa lutein cells, and theca interna cells are called theca lutein cells, which produce progesterone and estrogen respectively. It's written androstenedione, but later gets converted to estradiol and forms estrogen. Yeah. So what happens is, if there is no fertilization, the granulosa lutein cells and the theca lutein cells do not exist, because of which the progesterone level drops, which causes the blood vessels to spasm, and it causes the menstruation, right? However, if in if the fertilization does occur, up if fertilization does occur, corpus luteum acts as a temporary endocrine gland and secretes progesterone to ensure that the zygote can attach to the uterine wall. Yeah. So it helps in sustaining pregnancy. That's the main function of corpus luteum. So a key takeaway from corpus luteum is simply the two main cells, granulosa lutein cells, theca lutein cells. If in case of fertilization, uh, like as we know, these cells secrete progesterone and estrogen. It actually helps maintain pregnancy up until placenta is formed. And if the fertilization does not occur, the progesterone levels drop, which causes the blood vessels to spasm and results in menstruation. So that's that's about it. I hope it made sense. Thank you so much.